I'm Céline Arzel. I'm working at Turku University as an academic research fellow, and I'm also employed at OMBU. My project was focusing on endocrine-disrupting chemicals and their effects in waterbirds. So I will present here our research, past 5 years research results, and this project. Or let's say that the research is still ongoing. We have still many things, so it's only one part of our research. So why study endocrine disrupting chemicals in waterbirds? First, this came from observation that we noticed, or it has been noticed by many ornithologists, that there's a major decline in many waterbird species in Europe, and also an increase in the sex ratio bias towards males in bird censuses, but also in wing surveys, not only in Europe, but also in North America. In addition, in some colonies, which we are following, we have noticed a high rate of hatching failure. There are lots of eggs which are not hatching. Some have full developed embryos, some have partially developed embryos, so there are really different stages. So all those raised concern about endocrine disrupting chemicals may be affecting these populations. So what are the endocrine disrupting chemicals? An endocrine disruptor is an exogenous substance or mixture that alters function of the endocrine system and consequently will cause adverse health effects on an organism, but also on its progeny and the subpopulations. So the adverse effect will be a change in the morphology, physiology, growth, development, reproduction of an organism, but also of the system or subpopulation, which will be creating an impairment. It will affect the capacity of the individual to cope for stress. It will increase many issues and might have effect on the population dynamic. So endocrine disrupting chemicals, they can act as hormone mimics or blockers, but also they can also act on proteins, which will control the delivery of hormones to its target or the cell or tissue. So they have many different ways of acting and interacting with our bodies, but also with different organisms. They can be active at very low concentrations. They don't have to be there at high concentrations. They can interact with multiple hormones at the same time, and they can work together and have some additive or synergistic effect, meaning that it's a very complex system and it's very difficult to study these processes. So why would and how would endocrine disrupting chemicals affect the birds? Well, first, you should know that the sex differentiation in birds is very dynamic. It's a mix of genetic and hormonal processes. So depending how the eggs are taken care of, it will affect the male or female ratio in the eggs. Also, the males are the homomorphic sex, while the females are the heteromorphic sex, meaning that the characteristics of a female are very much driven by hormones, and a female bird can actually become a male bird under specific conditions and be able to reproduce as a male bird. But endocrine disrupting chemicals, they can not only affect the eggs, also this process of female becoming male, but they can also affect the survival or the fitness or the survival, let's say mortality risk for the females, also by affecting their metabolism and their immune process. And endocrine disrupting chemicals, we are exposed to them on a daily basis. We are releasing them. We are exposed to bisphenols, for example, from canned food, PFAS from cardboard, from pizza, for example, phthalates from electric cables, parabens from cosmetics, benzophenanes from sunscreen, flame retardants from all our electronics. So on a daily basis, we are exposed to a cocktail of contaminants, of these endocrine disrupting chemicals, but we are also releasing them in the environment on a daily basis. There are over 350,000 chemicals in the global chemical market. So there's a huge amount of chemicals out there, which is a huge revenue. 4.3 trillion US dollars, that was in 2017, so probably much more now. And the production, not the consumption, just one share of it, the production of chemicals considered hazardous to health in Europe in 2016, again, that's a bit old, was 200 million tons. So we can expect even, it's not only the production, but it's also the consumption, which is a problem. Because chemicals are not only released at the site where they are produced, so they will be released by in the atmosphere by particles, atmospheric, they will go in the atmosphere. And they are also released when we are using them. For example, fire retardants, when there's a fire in the forest and the firefighters are coming to extinguish it, then they are sometimes using fire retardants and it will stay in the environment. Also, when we are using soap and shampoo, some other products which contain parabens, for example, then it's released in the sewage water treatment system, and they are not adapted to collect those, so they will end up in the environment. Also, of course, with agricultural practices, we are releasing lots of biocides, and so also at the site of production with the industrial discharges, those are also being released in the environment. And often all those chemicals, when they are released in the environment, they will end up in the water environment. So they will end up in the sea, in the lakes, and the animals which are living in those habitats will be in contact with those contaminants, either via the food chain or directly via contact of contaminated water. And also, it's not only the area where the contaminants are produced that are the issue, it's also that there are long-range atmospheric effects, or it's not only at the place where they are released that they will be found very far away. For example, this grasshopper effect is very well known, that the chemicals evaporate with the warm air, and they will return to Earth with rain and snow in colder areas of the globe, which is why the Arctic area is extremely polluted, even if they are not producing or even consuming so many chemicals. So on the right, there's this kind of time scale of the atmospheric transport in the lowest part of the atmosphere. And you can see that if you release a contaminant or chemical in North America, for example, it will take 1 to 2 months to go to the Arctic, will take 2 weeks to go to Japan. All of that just to tell you that there is no pristine environment left on Earth. Where we release some chemicals, it will not stay there. It might go very far away, which means also that when we want to study contaminants, we need to keep that in mind, that we should not only focus on the ones that are produced in the area, but really screen for large number of contaminants. So our target species in our study are the common eider and the common goldeneye. For the common eider, we focused on some colonies in the south of Finland, while for the common goldeneye, we collected samples from all over Finland. For the common eider, we studied a small population on a small colony at Bengtsjar, which is a tiny island in the outer archipelago along the Finnish coast. It's so far from the mainland that there are no terrestrial predators. And because it's a touristic place, there are human activities which are acting as a scarecrow. So they are pushing away any avian predators. So on this tiny island, we have 500 breeding females. From this population, we realized that there was a very high level of unhatched eggs. We reached 43% of egg failure. And so we were wondering what was going on. So we decided to launch a study on this island. The females are there's lots of, they are breeding very much next to one another, and they are very tame. So here is a short video just to show you how tame they are and how we are doing our fieldwork.
Yes, it's funny.
So they are very tame on the island. So it's we are just picking up the bird, and then we are collecting blood sample from those females. And it's a very, very fast process. So we are doing it as fast as possible to decrease any kind of stress for the female, of course. And then we have collected blood from those females, and also we have collected 10 clutches from that island. We checked trace elements, and among the different trace elements which we investigated, lead was one with which showed the most scary result, let's say. So we had a very high level of lead in the blood, in the plasma from these females. So one female had concentration 10 times higher than the maximum mean values reported in the literature. It was really, really high. And the values were twice the concentration found in the Finnish colony, very nearby Finnish colony 10 years ago. So our lead level are really, really high. And we had the highest ever recorded lead level in the eggs also. At a lower level than that, there was an experiment done in Spain with farmed mallard, and they show that with even lower level than that, it would affect the survival of the ducklings after hatching, during the 7 days after hatching. So we can expect that its lead level will affect at least the survival of the duckling in our colony, at least. Then we investigate contaminants. We investigated contaminants of emerging concern also in the same colony. This has been published. And what we mean by contaminants of emerging concern, here we have a specific definition for our study. It's contaminants for which we have little or no knowledge about their environmental distribution, fate, and toxicities, but where we have some cause of concern. Often they are not regulated, and they are cause of concern because while they are not considered as being persistent, the fact that we are using them so much and releasing them so much on a daily basis in the environment, they are considered pseudo-persistent. They will be there, and the animals will be in contact with them. So we investigate 56 compounds from 5 groups. Phthalates and phthalate metabolites, so those are the ones which you find in electric cables. Bisphenols, you will find them in tap water, but also in plastics. Very famous bisphenol A, which was removed from baby bottles, for example. Benzophenanes are found in sunscreen. Benzothiazoles are contaminants which are used, or chemicals which are used for the wings of the plane in winter to prevent them from freezing. And parabens are often found in cosmetics, for example. So from our research on ADER from the plasma, we found that they are exposed to a cocktail of those contaminants. We find quite many of these contaminants in the females. Then we investigated if there was a transfer of those contaminants from the female to their eggs. And we can say, yes, there is a transfer. We found 17 of these contaminants in the eggs. We checked also the egg laying order to check if there was a difference between the first 8 legs as compared to the latest laid eggs. There was no difference. They were all they had all similar level of contaminants, which is good in terms of monitoring. It means also that we can just grab one egg and from a clutch instead of collecting the whole clutch to assess the contaminant level of that clutch for those contaminants. It's not the same for all contaminants, but at least for those contaminants which we investigated. What we noticed is that there's a difference between the plasma and the eggs. There are some contaminants which you will find only in the females, some contaminants which you will find only in the eggs, and quite many which you will find also in both. So just to say that it depends what you want to screen, and it's important to think what you want to screen before, if some of you want to do this kind of research, because you will have different results if you focus on the eggs or on the females. But interestingly, there is a transfer of these contaminants to the eggs, and we have no clue about their effects on the embryo development. We also checked the pair and polyfluoroalkyl substances, which are quite well known nowadays, and there has been lots of information about them in the newspaper. So we checked 41 of those PFAS in the either, both in the plasma and in the eggs. We found, or we detected, doesn't mean that there was not more, but at least we were able to detect 20 in the plasma, among which a PFD-ODS, which had never been detected earlier in either. We also detected PFAS in the eggs. We detected 18 of these PFAS and potentially an effect on sex hormone precursor, sorry. We investigated, because we had such a high level and such a broad amount of PFAS, we investigated if we would find the same in the common golden eye. So we found PFAS, 21 PFAS were detected out of the 41 screened. And the main area where we found at least the highest level of PFAS was Helsinki, so most urbanized of all our 5 locations. Second was Turku, which is southwest of Finland along the coastline. Then Kuopio, and then Lapland and Seinäjoki. So those 3 last one were quite equal with lowest level, but the 2, Helsinki and Turku, were the highest, the areas with the highest concentration of PFAS. So because of those results, we decided to conduct an in novo experiment on farm mallard eggs in order to determine if this level of PFAS might affect the development of the embryos. We conducted the first experiment in 2023. We injected the eggs and we euthanized the ducklings at hatching. We injected PFOS, which is a regulated PFAS, at a low dose and high dose, and we injected 2 other PFAS which are non-regulated, PFD-ODS and PFEX, and only at low doses for all of them. For low doses, those were doses which we found in the bird, so they are environmentally relevant concentrations. No, in the egg, sorry, not in the bird. The high doses were only for PFOS, and we had 149 eggs for this experiment. What we found is that we found an effect on the, well, let's say the first result was that we had a very high, that the eggs were not hatching, a high failure of eggs, which was probably due to the injection by itself. So out of the 149 eggs, we had only 44 eggs which hatched, which was very low level. But what we could see from those eggs was first that there was still an effect, at least from PFEX, on this hatching success. And we investigated also the steroid hormones from the ducklings. Out of the 18 steroid hormones which we investigated, we could detect 3. And for corticosterone, we had higher corticosterone plasma concentration in the ducklings exposed to PFD-ODS and PFOS high doses. As a reminder, PFOS high doses, or PFOS, is a regulated PFAS. PFD-ODS is not regulated. So what it shows here is that the non-regulated PFAS, is PFD-ODS, has as much effect, if not equivalent effect, to the regulated PFOS, which raised concern about this regulation and if those PFAS should also be regulated due to their effect. We conducted also a gene expression experiment on these ducklings. So we checked the gene expression in the liver. We saw a sex-specific change in genes related to metabolism and immune function. So we saw a disruption in the metabolic pathways, only in females exposed to PFD-ODS. That could affect their capacity to store fat, but also their seasonal behavior, their reproductive success, and overall their fitness and survival. We also noticed a disruption in antiviral gene expression in males exposed to PFEX. In the herd, we did not find any difference between those groups, but we saw a trend in the altered expression of genes involved in oxidative and cellular stress. So the results were not so clear. I will come back to that. And in the bursa of Fabricius, we noticed also an effect on the expression of genes involved in pathogen recognition and antiviral response. So from this, we could say that, okay, these PFAS, they seem to affect the metabolism and the immune function of birds, and it seems that there is a difference between males and females. We were not sure about the oxidative stress, so we conducted yet another experiment with from the same birds about the oxidative stress. And here we did not find any significant difference between the groups, which is good. So those PFAS, they did not seem to affect at least the oxidative stress in those ducklings. Because of our results, we decided to conduct another experiment in 2025. We, this time, kept the ducklings a bit longer. We euthanized them at 25 days. Why we did that? Because we wanted to investigate their immunological response, because we saw in the previous experiments that there might be something going on. And also we wanted to assess their behavior and if the PFAS might affect their behavior. In that experiment, we injected PFOS and two other non-regulated PFAS, PF Wunda and PFEX, and their mixture. This, I don't have yet the result because we just conducted it this year. In addition, we also had an experiment in which we used loggers, which we had in the eggs. So we had these egg loggers for common goldeneye and also for common eider. These loggers, they are recording the movement. So when the female is turning the egg, it will record that movement, that rotation, and it also records the temperature. Farshad is doing his PhD on the topic, and these are the egg loggers which we used for the common eider. So in that project, what we aim to study is if the combined effect of PFAS and other stressors are affecting the hatching success, also if there's a link with the hormones from the females, and if we see some altered hatching success. So from this study, the first result is that first, we detected PFAS, both in common goldeneye and in common eider. We found opposite result actually between common eider and common goldeneye. We found a positive correlation with egg turning rates and PFAS in common goldeneye, but we found a negative correlation with egg temperature in common eider. And there was a positive correlation with egg temperature in common goldeneye. But in both cases, there was no robust correlation between the PFAS and the thyroid hormones. And there was no also correlation between the PFAS and the hatching success. So the PFAS are affecting the way the females are attending the nests and their eggs, but it doesn't seem to affect the hatching success. So we don't know yet if it has enough effect on the embryo development, for example. So in conclusion, for the PFAS, the waterbirds are exposed to a cocktail of PFAS, among which the regulated one, but lots of non-regulated ones. There is a maternal transfer of PFAS. The effects of this PFAS on waterbird population trajectory need yet to be investigated. What we can conclude from now from this first in vivo experiment which we did is that at least those two unregulated PFAS, they have effects at least on hatching success and on the steroid hormone levels that are similar to the regulated PFAS, which raised some concern because they were used as low doses, and for the PFAS, this equivalent level is at a high dose. So they might have a lower toxicity threshold than the regulated PFAS. So we are investigating other endpoints at the moment. One good news is that we checked also neonicotinoids, which are these famous contaminants called the bee-killing contaminants. They have been regulated in Finland since 2013. They are still used, but in some very limited use, like, for example, veterinary use, like for medicine against ticks and fleas, and those chemicals are used, and some very limited other use. So we checked those contaminants and their metabolites at different locations all over Finland. We did not find any in our samples. So it can be also that our level of detection was too high to detect them. That's possible. Or then that they were not in the environment. Or that the trophic chain of the golden eye doesn't bioaccumulate those contaminants. So in summary, are waterbirds contaminated by potential endocrine disrupting chemicals? Yes, we observed that. Do these endocrine disrupting chemicals have biological effects on waterbirds? Yes, they have both effects on hormones, immune system, and their behavior. Is there a difference in the response depending on the sex? Yes. Can it explain the sex ratio biases and the decline of the population? We don't know, but at least it may contribute to it. How do birds bioaccumulate those contaminants? Well, that's another story. We don't know yet, but we have launched a ChemWet project last year. So that's a postdoc working in my team, Maria. She is conducting this project in which we are investigating the trophic chain of the common golden eye. So there will be more results about that later. So it's important to have a holistic approach here and to consider that it's not only that the birds are under pressure from the contaminants, they are under multiple pressures. They are, and especially migratory birds, they will be under pressure all along their flyway. So we conducted this, or we wrote this editorial 2 years ago in which we addressed this problem of these multiple stressors, because when you consider only one aspect, you might not have or find very significant result. But if you combine different stressors, you might see actually some strong effect. So it's important to think that there's not only contamination, there's also disturbance, climate change, habitat alteration, and many other things which might affect the birds. Je vais passer en français. So a small summary in French now. Donc, notre projet a porté sur les perturbateurs endocriniens et leurs effets chez les oiseaux d'eau. Nous sommes focalisés sur les éders à duvet, sur les garrots à œil d'or. Les deux espèces ont été étudiées en Finlande. Nous avons étudié pour les éders à duvet plusieurs contaminants, dont les éléments traces. Parmi eux, le plomb a été celui ayant des effets les plus notables et des niveaux, des concentrations parmi les plus élevés. Nous avons trouvé donc chez une femelle des niveaux 10 fois supérieurs à ce qui a été trouvé en moyenne. Et les niveaux pour le plomb trouvés dans les œufs étaient les niveaux les plus élevés qu'on ait jamais enregistrés jusqu'à présent. Nous avons également trouvé différents cocktails de contaminants émergents, ainsi que des PFAS, dont le PFDODS, qui n'avait été jamais rapporté jusque-là chez les éders. Et c'est un PFAS qui n'est pas régulé. Donc, ses effets sur l'avifaune n'est pas connu. Nous avons donc également étudié les PFAS chez les garrots à œil d'or. Nous avons regardé pour l'instant uniquement dans le plasma. Les études sur les œufs sont en cours. Donc, pour le plasma, nous avons trouvé les plus hauts niveaux de PFAS dans la zone d'Helsinki. En deuxième venait la zone de Turku. Les 3 autres zones qui sont plus au nord étaient moins affectées par les PFAS. Donc, c'est essentiellement dans la zone côtière que nous avions les plus hauts niveaux de PFAS. Au vu de nos résultats et des fortes concentrations en PFAS, nous avons décidé de réaliser une expérience d'injection de PFAS dans les œufs de canard colvert. En 2023, nous avons euthanasié les canetons à l'éclosion et nous avions injecté 3 différents types de PFAS, dont un régulé, 2 non régulés. Et tous étaient des PFAS que nous avions détectés dans les œufs de l'avifaune sauvage. Sur l'expérience de 2023, nous avons vu des effets sur le métabolisme, potentiellement sur la réponse immunitaire et aussi sur l'éclosion. En 2025, nous avons décidé de poursuivre l'étude et d'euthanasier les canetons à 35 jours, afin de pouvoir étudier l'effet sur leur comportement et plus précisément sur la réponse immunitaire également. Nous avons également une étude sur le comportement du retournement des œufs par les femelles d'Eider, pour lesquelles nous utilisons des systèmes embarqués dans des œufs avec des enregistreurs de rotation, donc de mouvement de l'œuf. Donc on enregistre à chaque fois que la femelle va tourner l'œuf et ça enregistre également la température dans le nid, ce qui nous permet donc de voir comment la femelle s'occupe des œufs. Nous avons noté un effet différent d'épiphase en fonction de l'espèce étudiée, avec un effet positif pour la température des œufs chez les garrots et négatif chez les éders. Et toutefois, les résultats, la contamination épiphase ne semblait pas affecter le succès d'éclosion ni pour les garrots ni pour les éders. Donc on ne sait pas, par contre, est-ce qu'il peut y avoir un effet sur le développement embryonnaire, mais qui n'affecterait pas l'éclosion. Ça reste à être étudié. Donc en résumé, les oiseaux d'eau sont-ils contaminés par les perturbateurs endocriniens? Oui. Ces perturbateurs endocriniens ont-ils des effets biologiques sur les oiseaux d'eau? Oui. À la fois au niveau des hormones, du système immunitaire et d'un point de vue comportemental. Et nous sommes en train d'aller plus loin avec l'étude en cours. Y a-t-il une différence dans la réponse selon le sexe? Oui, nous l'avons observé. Cela peut-il expliquer les déséquilibres de sexe ratio et le déclin des populations? Nous ne le savons pas à ce stade, mais cela pourrait au moins y contribuer. Et comment les oiseaux bioaccumulent-ils les contaminants? Nous ne le savons pas non plus, mais nous avons développé un projet qui a démarré l'année dernière, où nous étudions l'accumulation des PFAS dans la chaîne alimentaire du garrot à œil d'or. Toutefois, il est important de considérer tout cela dans son ensemble et d'avoir une approche holistique lorsqu'on s'occupe de la conservation, de la gestion de l'avifaune, notamment migratrice, parce qu'ils subissent non seulement les pressions par les contaminants, mais bien d'autres pressions au niveau de leur voie de migration. So, thank you so much. I would like to thank now my funders, but also all the students and all the collaborators, among which the bird ringers who have been helping a lot in the field work. It's been a huge amount of work, and it would not have been achieved without a great team. Thank you.